Assalamualaikum warahmatullahi wabarakatuh. We are from group 9. We will present about utilization of biotechnology in the control of viruses causing plant disease. Before we explain it, let's introduce ourselves. My name is Luhung Bhakti, number 188.
My name is Teksenia Saswati with student number 189.
My name is Abni Jelang Milani Syuhadha with student number 190.
My name is Rustiana Maharani with student number 192.
And the last, my name is Syifa Fairuz Zahra with student number 194.
This is our mind maps. First, we will explain about biotechnology. Second, about plants virus. Third, disease in plants. Fourth, hands of biotechnology in control of viruses that cause disease in plants. And fifth is example of utilization of biotechnology in control of disease causing viruses in plants. Biotechnology. Biotechnology is a combination of biochemical, microbiological, and engineering science to produce processes, products, or services that can be utilized for humans. Biotechnology has the potential to increase agricultural production, namely by improving crop yields and nutritional value, expanding crop production into hostile habitats, and improving crop protection against pests. Biotechnology in the field of plant protection includes the use or changing the genetic characteristic of viruses, microorganisms, either plants or animal cells, cell parts, and their metabolic products to control plant destroyers.
Next is plant virus. Viruses are minuscule agents visible only through an electron microscope and reproduce only in living cells. Plant virus is actually a simple macromolecule between 20 until 100 nanometer in size, which is passive when it enters the cell of the host plant it infects. Viruses consist of ribonucleic acid or ribonucleic acid or RNA or deoxyribonucleic acid or DNA covered by a protein coat or lipoprotein. Based on research, it can be seen that first, viruses can consist of various strains which can cause very different symptoms in the same host plant. Second, different viruses, their collective species, can cause the same symptoms in the same host plant. Third, some disease can be caused by a mixture of viruses that have nothing to do with each other. Next. Some examples of viruses that cause disease in plants are: first, tobacco mosaic virus or TMV is a virus that cause mosaic disease in tobacco plants. Second, rice tungro bacilliform virus or RTBV is a virus that causes tungro disease in rice plants. Third, tomato spotted wilt virus or TSWV is a virus that causes spotted wilt disease in tomato plants and various other plants. Fourth, tomato yellow leaf curl virus or TYLCV is a virus that causes curly jaundice in tomato plants. 5. Cucumber mosaic virus or CMV is a virus that causes mosaic disease in cucumber and other plant species. Next.
Disease in plants. Disease is an abnormal and detrimental physiological process of plant caused by primary factor, biotic or abiotic, and the disturbance is continuous and the result is indicated by abnormal cell or tissue activity. The occurrence of disease is largely determined by the interaction of three factors known as the disease triangle, namely, 1. The host or plant is which the pathogen obtains food or its necessities of life. 2. Pathogens are organisms that cause disease. 3. The environment, especially those that are abiotic, temperature, humidity, rainfall, wind, sunshine intensity. Next, kinds of biotechnology in control of viruses that cause disease in plants. 1. Plant tissue culture is resistant to disease. Tissue culture is a technique to isolate and maintain tissue pieces or plant parts explant on the appropriate artificial medium. Based on the explant material used, there are several tissue culture techniques to obtain disease-resistant plants, namely meristem culture, callus tissue culture, cell culture, and isolation of disease-resistant mutants from plant cells culture. 2. Protoplasm fusion. Protoplasm is a plant cell without a cell wall or only plasmodium so that genetic manipulation can be easier. Protoplast fusion will facilitate the incorporation between two lots of disease-resistant genes of different species or closely related.
And the number 3 is undead culture. Undead culture is a technique of shortening the crossing cycle or hybridization and producing stable homozygous double haploid plants. And the number 4 is embryo rescue or immature embryo culture. Immature embryo culture is done by implementing immature embryos to save the embryo from dying. And number 5, and the last is genetic engineering. The plant genetic engineering can be said to be an attempt to transfer certain genes or DNA from one plant species to another, both close relative and distant relative, or the search of these genes can be from other organisms such as fungi, bacteria, and viruses, and it can express it in the receiving species. Genetic engineering can be done by cloning genes and transforming genes into plant. Next. This is the example of utilization of biotechnology in control of diseasing causing virus in plant. The first one is Tungro disease in a rice plant that caused by RTB virus and RTC virus, and also mosaic disease in chili plant that caused by CM virus. Next. The first one, we are going to talk about biotechnology in controlling the Tungro virus. Tungro is caused by multiple infection of two different viruses, namely rice Tungro bacillarium virus or RTBV and rice Tungro spherical virus or RTSV, both of which are transmitted semi-persistently by the green plantoper Neptorix virescens. The development of the recombinant DNA technology opens up opportunity for assembling tumoroviruses-resistant plants through genetic engineering. Based on the pathogenicity factor of tumorovirus and tumor disease, it is hoped that the assembly of tumorovirus-resistant transgenic varieties can be assembled through transformation technology. The assembly of transgenic varieties can be done by transferring gen from viruses to plant, which are commonly known as pathogen-derived resistant or PDR. There are two transformation techniques that are commonly used, namely direct transformation through agrobacterium tumefaciens with microinjection, electroporation, and particle shooting. Transformation system with a tumefaciens is more widely used because it is relatively cheaper, efficient, and stable in introduction can. Transformation will produce putative transgenic test material. It is possible that kinetic putative will be obtained with various levels of resistance, namely sensitive, resistant, very resistant, and immune. After that, gain expression analysis and segregation analysis were carried out. Furthermore, it is necessary to improve transgenic plant through crossing transgenic plant with lines that have good agronomic properties. Next.
The second is biotechnology in controlling CMV. Cucumber mosaic virus or CMV belongs to the Cacaomovirus group. This virus has the widest host range among plant virus known to date. Is reported to infect more than 8,000 plant species and can cause major losses to chili plants. Satellite RNA or SAT-RNA is a small linear single-stranded RNA molecule that functions or acts as a parasite of certain viral RNA. Several SAT-RNA variants have been reported to affect the symptoms of CMV-induced disease such as SAT-RNA-D, -VL1, and etc. Cross-protection using SAT-RNA can be used to suppress the CMV virus. In participle, cross-protection is the use of a weak isolate of a virus to protect plants from economic damage caused by infection with the same viral isolate. Transgenic plants with lines that have good agronomic properties. The cross-protection mechanism occurs at the beginning of the interaction in plants between the protecting virus containing SAT-RNA and the virus the infect later, or the challenge virus, or during replication of the later infecting virus. If cross-protection occurs the beginning of the interaction, the later infecting virus or the challenge virus is inhibited by the process of releasing its coat protein or uncoating, so that the infection, the incitation of its replication can be prevented. From our discussion, the conclusion we can have based on above that, it can be concluded that biotechnology has combination of biochemical, microbiological, and engineering science to produce process, product, or service that can be utilized for humans. One of that use biotechnology in agriculture is to control viruses that cause germs in plants. Viruses are very small, again, visible only through an electron microscope and produced only in living cells. Whereas disease is an abnormal and detrimental psychological process of plants caused by primary factors, such as biotic and abiotic. And the disturbance is continuous, and the result is expressed by abnormal cells or tissue activity. Biotechnology techniques that can be applied include culture of resistant plant tissue, protoplasmic fusion, other culture, embryo rescue, and genetic engineering technique. The use of biotechnology is controlled by RTBV and RSTV virus that cause tumor disease is through genetic engineering to create transgenic plants that have a good agronomic properties. In mosaic disease caused by the CMV virus, cross-protection can be done using satellite RNA or SAT-RNA. That is from our group. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.